Hello friends, welcome back to my channel. In this video lecture, we will be talking about the three modes of RNA processing in eukaryotes. You know, RNA processing is very much different in both prokaryotes and eukaryotes. It is much complicated in eukaryotes compared to the prokaryotes because prokaryotic DNA is present in the cytosol as they don't bear any defined nucleus, and so the RNA is also made in the cytosol. And the RNA that is made in the prokaryotes, they carry all the necessary genes that makes the protein. But in eukaryotes, the transcription takes place inside the nucleus, and when the RNA is made from the DNA, the RNA is immature and it contains basically two different types of units. One is known as the introns and the other is known as the EXons. EXons are the coding element, while introns don't code for any protein. They are just the junk elements. So they need to cut all those introns out and join the rest of the EXons together. And this process is known as splicing. After the splicing, the mRNA is called a matured mRNA. But before splicing, there are two more important processes that should be carried out to finally transport the mRNA into the cytosol. So before transporting the newly made mRNA to the cytosol in eukaryotes, three major steps occurs. One is known as the 5' modification, or also known as the 5' capping. That is the addition of guanine residues in the 5' terminal of the mRNA. Second is the 3' adenylation, where repeated adenine residues are being added one after the another in the 3' section of the newly made mRNA. And then the splicing are carried out when the introns are cut out while the EXons are joined together. And that is known as the mature mRNA, and that is ready for translation into the cytosol. Now let's understand what is 5' capping. Basically, when you see the 5' terminal, we have three phosphate groups attached to the RNA. Okay, so when capping starts, they get involved with the enzyme known as triphosphatase. The significance of this enzyme is that it cleaves one phosphate group from the 5' terminal. So now after this cleavage, what we have is the structure like this with one less phosphate group from this 5' end. After that, there comes another enzyme known as the Guanil Transference. The purpose of this enzyme is that it takes guanine residues and put it and link it with one of the phosphate group in an inverted form. And so now the rest of the phosphate will appear as this. And in this process they require GTP, and pyrophosphate will be released because guanine monophosphate is taken and is added to the 5' end. And then there is some further modification, that is methylation. This methylation is present in some species, but in other species there may be no methylation, okay, no methylation event. During methylation, it simply adds the S-adenosyl methionine, and they simply transfer the methyl group to the 7th position in the base. And in some species there may be multiple methylation event. And the significance of this methylation event is that it acts as a cap, and this helps the mature mRNA to be guided into the cytosol from the nucleus. So this is all about 5' capping. In the next video, we will be discussing about the other types of mRNA modification. So I hope you like the video. So if you are new to my channel, do subscribe to my channel.